Okay, hello everyone, my name's Andrew Moyes and I'm a PhD student at Queen's University Belfast, alongside my collaborators Dr. Zhang, Professor Ji Ming, Dr. Zhou, and Professor Danny Crookes. I've been working on unsupervised deep learning for stain separation and artifact detection in histopathology images. Stain separation is a pre-processing technique which is commonly used in histopathology image analysis pipelines. Stain separation can enable stain color normalization between domains. In the left-hand figure here, the stain color appearance of images in domain A have been mapped to domain B, but the underlying tissue structure remains the same. This means that a machine learning model that was trained exclusively on images from domain B would be able to be applied to images from domain A. Stain separation can also make tasks like cell counting and classification easier. For example, by isolating the primarily hematoxylin absorbing nuclei, segmentation of cells becomes easier as irrelevant information has been discarded. Stain separation typically decomposes a tissue image into stain concentrations or densities, visualized here as black and white images. These represent how much of each stain has been absorbed at each pixel location. Alongside the stain concentrations, stain colors are also estimated. Here we can see that the colors have been determined for hematoxylin and eosin, which have been used to colorize the black and white images from the previous slide. The basic stain separation model is shown here. Essentially, a flattened tissue image X can be decomposed into a stain color matrix W and a stain concentration matrix H. Here N represents the number of pixels in the image X. Color deconvolution is one of the earliest known methods for stain separation. The stain color matrix W is determined by selecting tissue areas that contain primarily one stain and taking the average RGB value of these areas to be the stain color. For example, here pixels containing nuclei have been selected in green. The average of these pixels is the estimated color for hematoxylin. Once the color matrix W is determined, the stain concentration matrix H can be determined using a matrix inversion. However, in this work, we focus on an automatic method which utilizes sparse non-negative matrix factorization to estimate W and H. The objective for sparse non-negative matrix factorization is shown here. The terms highlighted in green represent the reconstruction loss, whilst those highlighted in purple represent a sparsity loss on the concentration values in H. Most existing methods make the assumption that the color of a stain is static across spatial locations in an image, regardless of the kind of tissue present at each location. This assumption dramatically reduces the computational complexity of the problem, and good results can be achieved when using it. However, in this work, we show that better results can be achieved when not using this assumption. On the left-hand figure, RGB pixels sampled randomly from an image containing hematoxylin only are plotted according to their respective intensity values. It can be seen that the colors vary in length and angle, meaning that the color of hematoxylin does vary at different locations in the image. On the right, these sampled RGB vectors are normalized to unit length, and principal component analysis is applied to visualize the color variance. It can be seen that hematoxylin can be expressed in a variety of colors from deep purple to rich blue. If we can include contextual information to better understand the kind of tissue at each spatial location in an image, better stain separation results can be achieved by modeling tissue-stain relationships. Another problem is that existing methods assume that tissue images are clean. However, artifacts are very common in tissue images such as carbon deposits, pen marks, muck, air bubbles, and dust. An example from the Cancer Genome Atlas is shown here. Blue pen marks are immediately obvious, and muck can be seen around tissue edges. These areas typically cause problems with stain separation. In this work, we primarily used the benchmark stain separation dataset publicly released by McCann et al. In 2014. This dataset contains 3 tissue specimens. Each specimen has been stained with either hematoxylin only, eosin only, or hematoxylin and eosin. The stained specimen is then digitally imaged, destained, and the process is repeated for the other stain combinations. This results in a dataset where each tissue specimen has been digitally imaged with hematoxylin only, eosin only, or both. Within our work, the images stained with both hematoxylin and eosin are used as input, and the single stained images are used to assess the quality of our proposed stain separation method. Here we show an overview of our proposed stain separation system. RGB tissue images are converted to the optical density space using the Beer-Lambert transform. Contextual information is captured using a U-Net-based convolutional neural network which produces a pixel-to-pixel feature mapping. These pixel-wise features are used to predict stain concentration or density values for each stain at each pixel location. Existing non-negative matrix factorization-based methods predict a stain color matrix W for the whole image. In this work, we utilize the power of convolutional neural networks to produce a stain color matrix W for each pixel separately. These pixel-wise concentrations and colors are combined to form a reconstruction of the input image. A reconstruction loss is calculated alongside a sparsity loss. This overall loss is minimized through backpropagation, and then our parameters are updated using the Adam optimizer. Here we show an overview of the U-Net-based feature extraction model. Image-level information is captured by the encoder. The decoder convolutions receive features from any previous decoder layers and corresponding layers in the encoder. This enables the fusion of high and low-level information. Here, the dashed green lines indicate skip connections via feature concatenation. As mentioned, in contrast to existing methods, we model the input images X with pixel-wise concentrations and pixel-wise colors, as opposed to pixel-wise concentrations and image-wise colors. Here we present some results of our proposed stain separation system. We compare our approach to a number of commonly used methods. Marchenko's method uses singular value decomposition. Farhadani's uses sparse non-negative matrix factorization, but with image-level colored matrices, and the color deconvolution method, which has been discussed previously. Comparisons are made by calculating the mean squared error between the reconstructed hematoxylin channels and the actual hematoxylin-only image. We can see in the table on the left that according to this measure, our approach was able to beat all other comparative methods. In the figure on the right-hand side, it becomes visually clear that our method achieves the most similar representation of hematoxylin only. The second aspect of our work focuses on artifact detection in hematoxylin and eSIN images. In Figure A, we can see the reconstruction produced by a non-negative matrix factorization-based stain separation method in the presence of artifacts. The artifacts have been mapped to the hematoxylin's purple. In a cell counting task, these artifacts would lead to inaccurate results. In Figure B, we can see that a different stain separation method has successfully separated the artifact from the tissue, which is beneficial. However, the stains in the tissue itself have not been separated at all. It is also important to note that in the bottom image in Figure B, the tissue has been reconstructed using a single colour, and thus information has been lost between the two stains. We propose an artifact detection system that can be used to detect and remove artifacts prior to the application of stain separation methods. Our proposed artifact detection approach is based on generative adversarial networks. The discriminator must learn a decision boundary between RGB colours that do occur in hematoxylin and eSIN images and those that do not. The generator is used to sample RGB colours that do not occur in hematoxylin and eSIN images. The discriminator assigns probability values to pixels, representing their likelihood of occurring in hematoxylin and eSIN images. These probability values can be used to detect artifacts. Here we show how the generator produces sample colours. As with most GAN generators, a latent variable Z is sampled. However, this latent variable Z is sampled from a three-dimensional uniform distribution between 0 and 1. This variable Z is then interpreted as an RGB colour pixel. The generator observes Z and transforms it by adding an update vector called delta. In the figure shown, Z1 is bright green, which does not occur in hematoxylin and eSIN images. Therefore, the delta is the 0 vector. Z2 is purple, which does occur in hematoxylin and eSIN images. The generator must therefore change Z2 so it becomes a colour that does not occur in hematoxylin and eSIN images. Thus, the delta is non-zero. The objective for the generator is shown on the left. We can see that the log term is minimized by generating samples that receive a low probability score from the discriminator. A sparsity constraint is applied to the delta values. This ensures that the samples produced by the generator remain as close to the uniform RGB distribution as possible. Without this constraint, the generator falls into a failure mode where it produces one or two RGB colours repeatedly, which is not useful for training the discriminator. The discriminator's objective is minimized by maximizing the score assigned to pixels sampled from real hematoxylin ECIN images, and minimizing the score given to samples produced by the generator. Here we show the output of the proposed artifact detection system. Columns 1 to 4 show clean segmentation of carbon-based artifacts. However, a variety of other artifacts have been detected as well. Excess stain has been detected in columns 5 and 6, whilst artifacts arising from air bubbles or dirt on the slide have been detected in columns 7 to 9. Finally, in column 10, a pen mark type artifact has been detected. We can visually see that our proposed model can detect a variety of artifacts. This information can be used to reduce the impact of artifacts on existing stain separation methods. Thank you for listening to this presentation. If you have any questions or comments, feel free to email me at the address shown at the bottom right hand side.